Well, welcome to episode 2 of the Magic Hour. I'm Paul Bevan, founder and CEO of Magic Valley. We're an Australian cultivated meat company. Joining me today,
I'm Andrew Leslett. I'm the head of research and development, and allegedly today I'm going to be explaining the science behind Magic Valley's technology.
Absolutely. So let's talk a little bit today, Andrew, about trying to demystify some of the science behind creating cultivated meat products, where we go from the farm to the lab to the table. And I guess on episode 1, we touched a little bit around how we got started in terms of the process, taking a skin sample from, in our case, Lucy the lamb, who continues to go on and live out her natural life in her flock, in her paddock. Now, just on that, I just want to touch on a little bit around, before we get started, how I made your life more difficult to begin with, which is, really got to do, I guess, not just with the business side of things, but the ethical approach we take to developing cultivated meat products. And so it was really important to me for us, not to be using, animal components in the process, creating a cultivated meat product where the goal is to remove animals from the supply chain really makes no sense to, be killing animals in that process. And so there's some products, I guess, that is traditionally used, one of them being fetal bovine serum, which is known as FBS. Now, without getting into the details, but this is the reality. Fetal bovine serum is obtained from the fetuses of pregnant cows when they're slaughtered in the animal agriculture process. So, to me, it makes no sense to be continuing to do that when we're trying to, create a product that is more ethically produced. And so, I guess one of the mandates that I gave to you and to the team, when we first started, and the whole time that we're working on this process, is to not be using any FBS, which is widely used not just in this space, but in the life sciences sector, and it's been traditionally used. But do you want to touch on a little bit about that as part of us getting started on today's topic? So the mandate I gave you is, for us not to be using, animal products, and particularly not using FBS in the process for what we're doing in terms of cultivating meat.
Yeah, thanks, Paul. So as a scientist, what you do when you're presented with a problem that you're trying to solve is what you often do is go to the published literature, the papers that are out there, to look for a method for somebody that's done this before, to see, okay, how do we take some cells from an animal, in this case Lucy the lamb, just take a really small scrape after getting animal ethics approval. And how do you do that without using any animal products, without using serum or other animal products? And when we looked in the literature to see how to do that, we couldn't get a recipe to do that because nobody had done it before, or at least nobody has published it before. So what we had to do is come up with some recipes and try some commercially available products that are already out there for human clinical use, to see whether and which of those different recipes for the media. So media is just the liquid that you grow the cells in a dish in the lab, would be able to support the growth of a really small scrape of skin, less than the size of the tip of your little finger, to grow in the absence of serum. And that's what we were able to show for the first time, that we could do with lamb cells. And so we were pretty happy with that. We then grew those cells up, expanded them in culture, and froze them down ready for the next step. You're probably thinking at this point, well, so those are the cells exactly that you use to grow up to make meat. And it's not exactly what we do, because if we just keep growing those cells, which are skin cells mostly made up of fibroblasts, content warning, science word, also some keratinocytes. If we just keep growing those cells, they're subject to a thing called the Hayflick limit, where they can only make copies of themselves or divide a certain number of times, and then they'll just stop growing. Much like as humans, we don't grow forever. We stop growing, and well, everybody knows what happens to humans when they get old. So in order to not have those cells get old and stop growing, what we do is we reprogram them and turn them back into stem cells. In this case, they're called induced pluripotent stem cells. Now, this is a technology that was developed in the 2000s by a really cool, groundbreaking scientist called Shinya Yamanaka in Kyoto in Japan. And this technology can be applied in all sorts of different ways, but one way that it can be applied is to take animal cells, so in our case from Lucy the lamb, to turn them into stem cells. So at this point you might be thinking, well, what's a stem cell? So at its most basic definition, a stem cell is a cell that's able to do two things. It's able to make copies of itself, and it's also able to turn into something else. So there's many different types of stem cells. So if you're reading something and it just says stem cells, you need to dig a little bit further to find out what type of stem cells that the piece of literature that you're reading is talking about. So what we're doing is making pluripotent stem cells, which just means cells that are able to turn back into any type of cell in the body that we came from. So all of our bodies are made up of cells, all animal bodies are made up of cells, all plants are made up of cells. And each cell in each of those organisms or plants carries the same DNA. So what we're doing is actually wiping some of the marks off of the DNA and resetting it to an early state, or its earlier states, where it has the potential to turn into any cell type. So we know how to grow those cells and to expand those cells, and they actually grow really quite quickly. They have a very fast, what we call doubling time, and that translates into less time in culture to make more mass. So in our context at Magic Valley, that means more mass for less money over less time. Which means more product, which is a great thing. So as well as being able to make copies of themselves, we can follow recipes or create recipes to push those cells along pathways to become the main component cells of meat, which are muscle, fat, and connective tissue. So they're really quite a powerful cell source that you can use, that all still have exactly the same DNA. They're not a genetically modified product, that you can create muscle, fat, and connective tissue and put together into a meat product.
Amazing. So this has previously been used in life sciences for other purposes, obviously, and obviously that's now, in recent years being adapted to create products for human consumption. It's probably good to, I know you touched on, it's probably good to give a couple of definitions. Obviously, you touched on media. I say media to, you and I say media all the time. People within the industry say media, and you talk to someone outside the industry and they're like, TV, newspaper, like what are you talking about? I know you mentioned briefly just before, but when we say media, do you want to give a little bit more information about what media is?
So all media is the broth or soup, if you like, that we put onto our cells to provide them with the nutrients the cells need to grow. So it's, instead of an animal eating food and it going through their stomach and going through their blood and circulatory system to put the energy, the sugars and the amino acids and all the proteins required to keep the cells growing, we put that into a fairly simple and defined and animal product free media to keep the cells alive, dividing, and we change that media, so we add different factors to it to push the cells to become the cell types we want, such as a muscle cell or a fat cell.
Yeah. So
was that a good enough definition?
yeah. So I guess just as part of the process, so we take the well, I mean, every company does it differently within the cultivated meat space, which makes this, a little bit complicated to explain. And a lot of people who've heard about cultivated meat but aren't familiar with the process, from many years ago, would be, familiar with the term FBS, and people would be, have questions around that potentially other companies are still using. So we'll just caveat this with, we can't speak for every company within the industry. We can certainly speak for what we're doing and potentially what other approaches there are. So so we take that small sample of cells. We culture it in media, as you've described. What is really a replication of what would happen, inside the animal's body in terms of nutrient transfer and those sorts of things. Which is I guess why companies or the process that has traditionally been used involves FBS, because obviously blood will be found inside an animal and
absolutely
carry those nutrients and all those sorts of things. So I guess that's one of the reasons why. And just to be clear, we don't use FBS. Just to make that point clear, we don't use FBS in our process. And it's a question we get asked a lot. A lot of people ask us that question, around the use of FBS or any animal components for that matter. We do get that question a lot as well.
And if you're interested in more about FBS, I've actually written a small blog on our website about why we don't use FBS. If you're interested, so www.magicvalley.com.au.
Absolutely, good point. Really good point. So I guess another question we get asked quite a lot in terms of the process is, so you take that initial sample of cells. And as you mentioned, do we just grow that up into the end product? Do you grow a whole other animal? Do you grow a sirloin, a drumstick, do you grow up whole parts? We get asked that question a lot. So as you've explained, we don't do that, and we reprogram those cells. So just to break it down, I guess, yes, we take that initial sample, which in our instance was a skin scraping.
Absolutely.
So what would happen if we didn't turn those cells into induced pluripotent stem cells? What could that skin sample become? Is it just skin?
Yeah, pretty much just skin. And there's some other, I don't want to get too complex, but there's some other cells in there that can become different layers of the skin.
Okay.
And different parts of maybe some nerve cells that supply the skin, or some blood vessel cells that are close to there. But
so I know that some of the processes that other companies use, which is to take a muscle biopsy, for example. So from a living animal, take a muscle biopsy, and they're using that to create cultivated meat. Can that muscle biopsy then just become muscle?
Yeah, pretty much. A muscle biopsy's the capacity for that is to a muscle stem cell makes muscle. So it is a stem cell, but it's not a pluripotent stem cell. So it divides a bit slower, and two, it can only turn into muscle. And it also doesn't keep dividing again and again and again. So you have to be pretty careful about making stocks and making sure you keep a lot of early stocks of those cells so that you can make enough to make it profitable.
So, and that's limited by that Hayflick limit you were talking about previously.
Exactly. Yeah.
And would if you're taking that approach, so you take a muscle biopsy, it's got a limited lifespan, for want of a better term, it can only become muscle. Would you have to do that with fat, for example, as well, or other?
Absolutely. If you start with a committed stem cell or an adult stem cell, we call that type of stem cell, so a fat cell or a fat stem cell or a muscle stem cell, you do have to take that type of approach.
So you would have to replicate that for every cell or tissue type you want in the end product, basically.
Exactly. And if you don't get that from the same animal, then they may have different they will have different DNA, and they may behave slightly differently. So you just need to be careful about keeping your product the same over time. and keeping your process consistent.
Yeah, absolutely, absolutely. I mean, something we haven't really spoken about, a lot of is the regulatory approval process in terms of creating a product, a process, and a product, for human consumption. And obviously, there's a lot of stringent guidelines, safety guidelines, around what we're doing, what all the companies in the industry are doing, and so that could be, as you pointed out, that could become problematic if you've got to replicate that process for all these different cell or tissue types, or if you have to change that, or if something goes wrong, I can foresee a whole lot of issues there.
Yeah, definitely. I totally agree with that, Paul. It's that's why we think the technology that we're using is the best way forward. And that's how we differentiate ourselves from other companies. So there's a lot of, I guess, misconceptions about what a cell-based meat product is, and especially the one at Magic Valley. It's not plant-based, so it's not made of plant. So it's actually going to have real meat in the product. And it's different from traditional meat in the ways, I think we talked about a fair bit in the first episode, where it's obviously there's no animals being killed, but it's not taking up a lot of agricultural land, which potentially frees up land for planting more trees and is better for the climate, better for the planet.
Absolutely. It's a point that often gets overlooked. I think a lot of people talk about the sustainability of the products, and cultivated meat has so many advantages. Another question I just wanted to ask you, I guess, around as well. Obviously, so we take that initial skin sample, we reprogram, is that the right word, to take it back to a pluripotent stem cell?
Yes, scientist Paul, it is.
Yes, thank you. I've learnt a little bit. So we take that back to an induced pluripotent stem cell, and then we can direct that induced pluripotent stem cell to become, as you mentioned, muscle, fat, connective tissue, which we obviously would combine to create an end product. So molecularly, it's the same?
Absolutely. It may not be identically the same. This is my scientist brain answering you, but it's really, really close in terms of the percentages of different proteins and fats, and it actually gives us a bit of an advantage because it allows us to add in a little bit more fat for flavour, maybe, if we wanted to, and taste, but also for health we can dial down the amount of fat that goes in. We can add in different
vitamins,
ingredients that we want to put in, such as omega-3, vitamin B12, for example.
Yeah.
So yeah, it gives us a lot of flexibility. Again, with the caveat that all of that has to be approved by the regulators before it becomes a product that is being eaten by the general public.
Absolutely. Another question we get asked a lot is around GMO. GMO. A lot of people think that all cultivated meat products will be GMO, but that's not the case.
No. So the process we're using is not GMO. The DNA in our start cells from Lucy the lamb is exactly the same as the DNA in the cells that we're produced our prototype burgers and tacos that you may have seen on our website from. So it's exactly the same DNA. We have not modified that DNA. Some companies are taking the approach of using genetic modifications to make their cells grow faster, to make their cells more quickly turn into other products. My worry about those approaches are that we don't exactly know how stable those approaches are over a very long term time, and how stable they are in growing large numbers of cells in large volumes, which is what's going to be required to produce meat products.
Absolutely. Scalability is one issue, but if you have to change your process, for example, once you've got a product or a process approved, that's a massive headache in terms of a business perspective, I guess. Probably it's a good time to touch on a bit more of the business benefits of using induced pluripotent stem cells. I know when I first came to you, with this idea that, we wanted to create cultivated meat, we had to be able to create cultivated meat at scale, because that was the only way that we were going to have, a large enough impact, because there's, without making it too simplistic, a lot of people or companies can create cultivated meat products in the lab, and create a burger. But that's not going to make any difference to anyone. It has to be able to be produced, at scale. And so I know you touched on briefly a little bit before around doubling time. So it's probably something we should cover off in terms of doubling time and creating mass and the lifespan of an induced pluripotent stem cell.
Yeah, so content warning, a little bit of maths. Doubling time is very simply the time it takes for one cell to become 2 cells and then to become 4 cells and 8 cells, etc. So it's a measure we use as scientists to work out how fast cells are growing, and it can change over time. And you need to keep an eye on it and monitor that. But it's quite well recognised that induced pluripotent stem cells have a faster doubling time than all of the cells that are in the body, and there's a reason for that. It makes sense in a developmental sense.
So much faster doubling time. They are indefinite, so no Hayflick limit on an induced.
No, so they actually there's at the end of chromosomes there's these things called telomeres. Sorry, this is heavy science now. And once you reprogram a cell, the telomerase activity is changed so that the telomeres do not shorten. So basically, these cells do not age if you keep them in the conditions that we know that they like and that we know that allows them to keep doubling and keep expanding.
Okay, that makes sense. So an induced pluripotent stem cell, it has a shorter doubling time and can multiply indefinitely.
Yep.
Yeah. So obviously from a business perspective, one of the main cost drivers is the media that is used. How often do you have to change the media in the cells when you're culturing them? Is it equivalent to the doubling time or something similar?
It's a little bit of a moving target with the different types of media that are being developed. The traditional change of media is on a daily basis. There's some new components to media that are a little bit more stable, that are allowing maybe every second day. And when you move from lab-based 2-dimensional culture of cells, so in like a petri dish, people are probably more familiar with, we often use different sized flasks that are made of plastic, but you can then move into machinery called bioreactors, which start at around a 50 mil volume and go up as high as 200,000 litres in volume. And that's where cells don't grow attached to a surface usually. They grow in what we call suspension, so floating as small aggregates in the media, which you now are all experts on because we've defined it.
Yeah, and those 200,000 litre bioreactors, they're still being designed and built.
They've been planned. Yeah.
Yeah, but theoretically in the modelling, then we absolutely can use those once they're constructed.
Exactly. Yeah. No, we do not have a Magic Valley 200,000 litre bioreactor yet.
We do not. Okay, so that's really clear. So an induced pluripotent stem cell, indefinite lifespan, a faster doubling time, less media required. So because you've got that faster doubling time, so to get to the end of that process. So that would mean from a business perspective, less cost, which would also mean a lower price point for the final product. I guess the point that we didn't touch on was that the pluripotent stem cell can become any cell or tissue type. So we can direct them to become muscle or fat or connective tissue. And that would be in the same bioreactor or in different bioreactors for each of those.
Our plan is to do that in different bioreactors. The media additives that you add to push the cells towards muscle won't push the cells towards fat. So you need to have them in separate bioreactors, but then you can put them back together at the end of the process on harvest and mold them into the shapes and sizes that you want.
For the end product, for the consumer.
Exactly.
Yeah. So I guess on the food aspects or the food science aspects of creating cultivated meat, so we're growing up muscle and fat and connective tissue in the lab and forming that together to create a real meat product because it is real meat. What else goes into that final product? What are some of the other considerations? I think fat. I think a lot of people would agree that the fat content provides a lot of flavour to that end product. What are the other considerations? I know we've mentioned the maturation of muscle fibers and those sorts of things. So there's obviously a number of parts that are important to the end consumer when you're trying to replicate a product, and that's obviously around the taste, the texture, and the mouthfeel. How would a cultivated meat company go about looking into those issues and trying to replicate it? Is it just trial and error initially until you get the balance right?
So obviously, it's a whole area of science that food scientists work. One of the aspects you left off, which is really quite important aspect, is the smell as well, and that is often associated with the different fats that are present in different types of meat, and how they're cooked can also affect the different types of fat.
I'm certainly not an expert in meat products, but I get what you're saying. Around the cooking and the caramelization of the fats and those sorts of things, which I think will be super important to the end consumer. Is there anything else you want to touch on around the end-to-end process of going from that skin scraping to creating the end product?
I think safety is one thing we need to be cognizant of always during this process, because we're growing large amounts of cells, or we're going to be growing large amounts of cells, and we need to be monitoring at every stage to make sure that there's nothing being introduced to the product that's going to be harmful at all. And we know exactly how to do that, and it's a matter of just putting all of the checks and balances in place, and that's exactly what the regulatory authorities are going to want to see. And we'll be working in a sterile, very controlled laboratory/factory environment with inline sensors in bioreactors to be able to detect any unwanted microorganisms, etc., or anything that's going wrong. So it really gives us a quite large amount of control over the safety of the product and the reproducibility of the product that comes out at the end, that isn't affected by weather, isn't affected by disease, isn't affected by would really potentially have a great supply chain reproducibility aspect to it.
There's a whole lot of big scary terms in there, but basically what you're saying is cultivated meat products will go through the same safety test that traditionally farmed meat products go through, that all food products go through, particularly when we're talking about Australia, with the regulators here, Food Standards Australia and New Zealand, which everyone shortens to FSANZ. And when you say FSANZ, it's like saying media and no one knows what you're talking about. But when we say FSANZ, we're talking about the regulator here for Australia and New Zealand. Do you want to talk a little bit about that process? We've obviously been in conversations, with FSANZ for a little while around introducing cultivated meat products to the end consumer and having them regulated.
Yeah, so in Australia, as Paul's just said, novel foods and in fact all food products are regulated by Food Standards Australia, New Zealand. So there's a really nice pathway called the Novel Food Pathway, where it's a 9-month or 9 to 12-month regulatory process where you go through and you have to address a whole range of criteria around what is in your product, how does it compare to other products, are there ingredients that have never been used before, what percentage are they in there, do they have allergy? There's a whole range of safety aspects to that process, and it's a really well regulated pathway for us to take once we are ready with our first product.
Absolutely. I'd probably go so far as to say, if let's see if you agree with me here, but I'd go so far as to say that developing cultivated meat products are probably even safer than traditionally farmed meat products because it's conducted in a sterile environment.
I would have no problem agreeing with that. I think that's entirely true.
Yeah. Just makes sense, really, doesn't it? I mean, some of the other benefits, I guess, obviously, of cultivated meat, when you compare it to conventional meat or traditionally farmed meat, really do relate back to the process. Whether it's safer, it's obviously definitely a cleaner environment. We have the ability, as you mentioned, within the lab to add certain nutritional additional components to enhance the nutritional profile. Removing saturated fat. Adding vitamin B12. Obviously, it's not something you can do with traditionally farmed products. They are what they are to an extent. Obviously, less. It's very topical. As we've seen over the past couple of years, in particular, in terms of foodborne illnesses as well. And another component I know you're really interested in and the benefits of is antimicrobial resistance. Do you want to talk about that a little bit?
Yeah. So again, for a little bit more detail, I've written a small blog on this on the Magic Valley website. But in a nutshell, there's something like 75% of the antibiotics produced worldwide are actually going into livestock. Now, as this happens, that they can pass through into people. And what happens is people around the world develop what's called antibiotic resistance, which means when they get sick, if they're given one type of antibiotic and they're resistant to it, they don't get better. Bacteria in them keep growing. So there's only a limited number of antibiotics that are available in the world. So if we can use less antibiotics, then that's a way of stopping the rise of antimicrobial resistance. And there's actually something like 1 million people die each year because of antimicrobial resistance. So it's a really important thing to be aware of, to not overuse antibiotics. And we talk about it a little bit in our kids and in humans, to not be prescribed antibiotics for something that is maybe a virus and not a bacteria that is what an antibiotic works on. But what we don't talk about a lot is the huge amount of antibiotic use that happens in livestock animals. And if we can reduce that, that's a really good thing.
Absolutely. That's a huge problem that I think probably most people aren't aware of.
100%.
Very interesting. All right. Is there anything else around the process that you'd like to mention before we sign off for this week?
No, I'd just like to thank you for making it as hard as possible without including animal components, because a challenge is always a good thing, and it's the ethically right thing to do.
I'm glad you think it's a challenge. That's excellent. That's not what you said early on, but that's okay. That's okay. All right. Well, I think that probably wraps us up this week for the Magic Hour. Please remember to follow us on the social channels. We're trying to build up our Instagram at the moment. Our Instagram handle, Andrew, is
Magic Valley 2020.
So please give us a follow. Also, head to the website magicvalley.com.au. You can subscribe to our mailing list there and keep up to date with everything that's happening with us. Thanks for listening, everyone. We'll be back shortly with episode 3.